Imagine a tool so precise it can rewrite the DNA inside your cells, fixing genetic diseases, modifying crops, and possibly creating designer babies. Welcome to the world of CRISPR. CRISPR isn't just some lab techniques. It's one of the biggest breakthrough in biology since the discovery of DNA. But what is CRISPR and how does it work like tiny molecular scissor? Let's break it down with me in my YouTube channel Bioscope. So the question is, what is CRISPR? CRISPR stands for clustered interspace short palindromic repeats. Biologists use the term to describe the genetic appearance of a system that was discovered in microbes, including bacteria and archaea, as early as 1987. For a long time, no one really understood what it did, but around 2005, researchers discovered CRISPR is an immune system. It's used by microbes to help protect themselves from invading viruses. To stop the invaders, the microbes use CRISPR to recognize and eliminate specific trespassers. Let's understand how the CRISPR work in a bacteria. In order for the bacterium to detect and eradicate the virus in subsequent infection, it integrates a portion of invader's DNA into its own genome when a virus or other intruder enters a bacterial cell. It is comparable to the immune system of human. We produce a large number of antibodies as an immune memory when we are infected by a virus. These antibodies then swiftly identify and eradicate the intruders when the same virus infects us once more. CRISPR aids in the establishment of a genetic memory. When a virus infects a bacterial cell, a fragment of virus's genome is taken by the bacteria, which then incorporates the DNA into its own. CRISPR uses newly obtained DNA sequence to generate a new guide RNA, which aids in the discovery of the invader through sequence complementary. For example, A binds to the T and C binds to the G. The guide RNA quickly identifies the virus's DNA sequence, attaches itself to it and eliminates it in the next time the virus infects the bacterial cell. Now meet Cas9, an enzyme that acts like tiny scissor. It teams up with guide RNA, a custom-made piece of code that tells Cas9 exactly where to cut. When Cas9 finds the matching sequence in the DNA, it cuts it and then cell rushes to repair the damage, just like fixing a typo. Scientists realize, hey, what if we use this system to edit any DNA we want? And yes, they did it. By designing custom guide RNA, we can tell Cas9 exactly where to cut. Once cut, the cell tries to repair the break and we can sneak into a new genetic information. So, in the CRISPR-Cas9 technique, we need a guide RNA or gRNA Cas9 enzyme. So, let's have a look how it actually works. So, this is the whole mechanism where we have a target DNA and accordingly which portion we have to cut, based on that we have to make a guide RNA, and this guide RNA knows exactly where they needs to bind. Then the Cas9 protein will come. For example, this is the target portion where the guide RNA will bind and then they will cut. After that cutting, we can delete a gene or we can insert a new gene to fix the break. Scientists use this moment to delete a faulty gene, to fix a mutation or to insert something completely new. CRISPR is already changing the world by curing diseases like sickle cell anemia. The first CRISPR drug, Cas9, used in the treatment of sickle cell anemia and beta thalassemia. In 2019, Victoria Gray was the first person in the US to receive CRISPR treatment for the genetic disease sickle cell anemia. Then we can use this technique to make pest resistant crops. We can use it to engineer microbes that can use as a biofuel or biomedicine. But with great power comes big question. Should we edit human embryos? What are the risks? And where do you draw the line? In 2018, a Chinese scientist edited human embryos, causing a global outcry. Because when we start editing human genes, where do we stop? Curing disease is one thing, but enhancing IQ or looks, that's where the debate gets real. Beyond this, some scientists may use this CRISPR technique for ecological reason. For example, eliminating mosquito. Every species exists for a reason. If we try to eliminate mosquitoes, we might invite our own destruction by disrupting a food chain followed by an ecosystem. CRISPR-Cas9 is rewriting the rules of biology. We are only beginning to understand its full potential and consequences. That's all for now. If you found this fascinating, hit like and subscribe for more such videos. Lastly, I want to ask you, should we use gene editing to make humans stronger or smarter? Share your ideas and opinions in the comments below. Thank you for watching.